Welcome back to our class. Today we're going to be talking about the paper two. This paper two was being written by um, one of the provinces. So we're going to be using it as a guideline for our September exam. Yes. However, we will try to highlight some of the areas where you can focus more related regarding to these upcoming papers. Also, especially for um, those provinces which have not written, and yeah, we can use it for final exams, and then we can also use it any exam as long as it has paper too. Anyway, let's start without wasting time. Is M.Saidi as usual. Thank you so much for subscribing, liking, sharing, taking care of this channel. This channel wouldn't have been there without you and without your input for following us. Yes, let's go to question number one, and then we see what is exactly they are talking about. Question number one. Which organelle represents the location of extra nuclear DNA in animal cell? When you talk about extra nuclear DNA, remember that we have three kinds of DNA. The first one is the nuclear DNA. The second one is the mitochondrial DNA. The third one is the chloroplastic DNA. So now, if you have the nuclear DNA, DNA code of life, basically here we are looking at the nuclear DNA. But we have other areas where you can find DNA in the cell. So if it is a plant cell, we can find it in the chloroplast. So we call it chloroplastic DNA. If it is animal cell, then we're going to find it in the mitochondrion. But because the mitochondrion can also be found in plant cell, then it means that in the plant cell, we can have the nuclear DNA, chloroplastic DNA, and the mitochondrion DNA. But animal cells only have the nuclear DNA and then the mitochondrial DNA. So what does it mean? What does it mean? It means that they said animal cell. If you look at it here, we see that they're talking only about the animal cell. So our focus is going to be on the cytoplasm out, nucleus is out, the mitochondrion, it can be found in plant cell and animal cell. Chloroplast is out. So because this is plant, this is no, this one they said extra nucleus. So the nucleus, they have talked about it. So the only answer which is much more correct is what? Is it C. So C becomes the most correct answer. So you get those 2 marks. We go to question 1.2. The main purpose of the genetic cross in the genetic crosstalk diagram is to measure the DNA sequence, not really determine the genetic mutation, not really predict the possibility of the offspring's characteristics, and then solve the what? Solve the crime. So if you look at it here, we see that one of the function, not sequence, we see that when you do the genetic cross, you find what we call the offspring. So the offspring's characteristics can be obtained. So when you do a genetic cross or a genetic diagram, you're able to identify the possibilities of the offspring's characteristics. Yes, so the answer becomes C, so C becomes the most correct answer. Which one of the following is the significance of DNA replication? We see that chromatin network condense, that's the description which happens in the during interphase, so this is out. Complementary pairing of the nitrogenous bases, yes, it happens, but that's not the function. This will lead to what? So this one goes out. Weak hydrogen bonds break, yes, they break, but it's a description of what happens in the DNA replication or in the DNA in the protein synthesis. But in this case, they're talking about DNA replication. So that's not an answer. So this one goes out. The most correct answer is the only option which is remaining, and they're saying that the genetic material is doubled. You double the genetic material so that when the cell divides, you're able when the cell divides, each cell must have equal amount of the genetic material. So that's the function of the DNA replication, so that it can bring about the multiplication of this DNA, so that during the division of the cell, the genetic material can be equal in each cell. So the answer becomes D. So D becomes the most correct answer, and then you get those 2 marks. Thank you so much for liking and subscribing. All right, we continue to the next point. The biological evolution refers, remember when you talk about evolution, it means that the increase in the complexity of an organism. So when you increase the complexity of an organism, so we call it you're evolving. Yes, but this, because it is about life, that's why you're linking it to an organism. So evolution can happen, evolution of a fawn, evolution of the sun, evolution of the moon, evolution of the what, but because it is about the organisms which have life, then it's going to become biological, because biology means, bio means life. So biological evolution must be referred to the increase in the complexity of an organism which has life. Let's go to the objectives and then we find out. Formation of new species, that's speciation. Gradual change in the characteristics of species, that is also known. Gradual change in the characteristics of a species. Over time, gradual change. You see, you are changing the characteristics. So this looks to be that correct. Present day distribution of species in the world, that is distribution of organisms, that is biogeography. Structures that have the same basic plan but adapt for different functions, though that is homologous structures. So this is also out. This is also out. So the most correct answer looks to be B. So B becomes the most correct answer, and you get your 2 marks. So we go to the next point. Peacock, you know them. Use bright colors to attract the mating. Mating, you know the mating. No, we don't need to explain about mating. Everyone knows about mating. Don't forget to stay virgin. All right. The reproductive isolation mechanism is called breeding, hybrid infertility. No, this is not about the colors. They're saying about reproduction at different times of the year. They're saying use bright colors, attract. There is nothing related to time there. So time of the year, no, out. Species specific courtship behavior. The way how they dance is not the same way you dance when you're looking for the mate. It's not the same way. So basically here, this one becomes the species specific courtship behavior. Each species has a specific way in which it attracts the different sex, the different gender. You understand? During the time of mating. So each species, when the frog is doing it's nice for them. It's ugly for you. But for the frogs, it's nice. So that's how they do their behavior. You understand? Yes. So this is species courtship behavior. Cooperation, you know that the insertion of the penis inside the vagina, that's out. It's not about that. So this is the most correct answer, which is species specific courtship behavior. Each courtship behavior is specific, a specific species. That's what it means. However, humans, some of them are ugly. You want to try everything. I didn't say that. So the answer is C, and then becomes the most correct answer. You get 2 marks. Thank you so much, guys. We continue. Life science is nice, guys. By the way. All right. Mendo, the scientist who played a role in the discovery of DNA. They are not talking about discover the DNA. They're asking the person who played a role. Whatever part you did take in the discovery of DNA, that's what they are looking for in this regard. Let's continue and find out. Mendo, and Crick. Yes, Crick, yes, but Mendo out. That is evolution. Bloom, but this evolution. Watson is yes, is correct, but Darwin, that is evolution, is out. Franklin, yes, correct, and Wilkins, yes, correct. So D becomes the most correct answer. James Watson, Francis Crick, Maurice Wilkins, and Rosalind Franklin. Those are the 4 individuals who were participating in the discovery of DNA structure based on our molecular. Yes, so that's it. We continue. Difference between genotype and phenotype is that the genotype is a genetic makeup, while phenotype is external. Genetic makeup, genotype, the gene type. Which kind of gene type do you have? Which kind of genetic composition do you have? That's the gene type. Is it a recessive? Is it dominant? Which type of gene you have? So that's what it means. And then you have the phenotype, the physical expression of an organism. How do I look? I'm dark-skinned, man. You see me. So that is physical expression of an organism. You see? So that is the phenotype. So this looks to be correct. Saying that the genotype is an allele inherited from maternal origin, while the phenotype is an allele from Norman. They're saying that the phenotype is external appearance. Norman, phenotype, gene type. So it's not about the external appearance. Genotype is the inheritance, while phenotype is not. Man, the characteristics you have, you obtain them from your father and your mother. So since it is like that, then it means that we inherit even the phenotype, because the genotype will determine the kind of the phenotype you have. And this genotype, you obtain it from the mother and the father. You understand? So, so this one also out. So the most correct answer becomes A, and A, you get 2 marks in that regard. You see, by the way, life science is so nice and enjoyable. When you try to do this life science, you'll find out that life science is nice. It's nice. By the way, every day you practice life science. How can you fail life science? Let's continue. 1.1.8. The graph below shows the average height of giraffe. Giraffes in different time period used by Lamarck in the evolution theory. All right, it's a story. The average height of giraffes in different time periods. Okay, sharp. Which one of the following is shows the correct percentage increase in the height of giraffe different times of the year or periods? Remember, percentage increase, we say that it is final minus initial over initial and times 100. So I need to find out that if I get the final, is saying that from 20 to 15, yeah, 20 to 15, yeah, 20 to 15, now means okay, from here to here. So if you put this minus this, divide by this, times 100, do you get this out? B, 15 to 10, meaning that final, which is 3.3 minus 2.2 divided by 2.2 times 100, do you get this? If you don't get it, then it's wrong. 10 to 5. 10 is 3.4, 3.3, and the 5 it is 4.5 minus 3.3 divided by 3.3 times 100. Do you get this? If no, this is wrong. Then 5 minus 1 or until present, 5 from here until present is 5.5 minus 4.1. That is 1 divided by 4.5. Then times 100. If you get this, then that becomes if it's wrong, then it's wrong. If you get it, then it means that that's the correct answer. So our most correct answer here is D. So D becomes the most correct answer. You understand? Yes. We go to the next question. 1.19. All right. The ultimate purpose of artificial selection is that increase the genetic variation. Artificial selection increase the genetic variation. I don't think so. Allows the organism to survive in the natural environment. Artificial? No, man. No, man. Reduce the size population of species by removing the organisms with the undesirable characteristics. Why should we reduce the size of the population? For the organisms, yet we want more of them. I don't think so. Let me see it. Increase the organism with the specific desirable trait for human benefit. Discard acid, for example, milk, different Characteristics we are looking in organism: meat, disease resistance, meat. I talk about meat because I like meat. Hey, man. Nyama. All right, we continue. 1.1.10. Which organic catalyst is used to cut the plasmid? Plasmid in genetic engineering. Remember, plasmid is that bacteria which, did I say bacteria? Yeah, is found in the bacteria. This plasmid is the one where the enzymes are used. We call them restriction enzymes, basically. We call them restriction enzymes. Restriction enzymes, these are enzymes which cut at a specific area, you understand, of the genetic material. So they are used to cut that plasmid's DNA so that the one which you want to multiply is inserted inside there. So basically, when they say the organic catalyst, definitely we are talking about the enzymes. There are specific, the enzymes, these ones, which we call the restriction enzymes. So they're saying enzymes, okay, looks to be correct. Stem cells, nope. Recombinant DNA technology, nope. Bacteria, nope. The plasmid is found in a bacteria, so it can't be. So the answer is A, which becomes the most correct answer. And that it means that you get 20 out of 20. So you see that, guys, it is so nice chilling, and getting 20 is very easy. Guys, make sure that in these objectives, you get 20 out of 20. Yes, if you see that you have challenge, check out what you think it's not right. There are some answers which you see that this is wrong, can't be. So the remaining one could be the correct answer. So you can use what you call the elimination method. Yes. All right, we got two question terminologies. I like them. I like them. Yes. Pattern of DNA bars, a pattern of bars, black bars, bands on X-ray films where sample is put through special biotech process. So we call these ones we call them DNA profile. These are DNA profile. Yes. Then they're saying that a fluid that has free RNA nucleotides complementary to DNA strand. A fluid that has DNA. When you say that fluid, remember, when you talk about free DNA, RNA nucleotides complementary to DNA strand, meaning that this, they are talking about, this is DNA. So where is DNA found? DNA is found in the nucleus. The what? In the nucleus. So if it's found in the nucleus, how do we call the fluid inside the nucleus? What is this fluid called? Since it's the fluid, the one which is outside here, we call it cytoplasm, the fluid of the cell. The one which is found inside, we call it nucleoplasm, the fluid of the nucleus. So the most correct answer here is going to become, we're going to say it is a nucleoplasm. The type of dominance that produce an intermediate phenotype. Whenever we talk about intermediate phenotypes, we're talking about incomplete dominancy. For example, none of the two is completely dominant over the other. It means that each contributes 50%. For example, I always give an example of presidents. The president of South Africa and the president of which country can I say? Zimbabwe, Malawi, Botswana, Eswatini. So all of those, if they are there, yes, they have the same power in their own countries. So when they sit on the table, they address them as presidents, meaning that if they are discussing about something, each vote will be equivalent to the same power. If this vote is one, even the other one is going to be one, meaning that they have equal power. None of them is completely dominant over the other if they are sitting on the same table. None of them None of them, because all of them are presidents. So if you have red crossed with the white, you produce a pink. Red is not completely dominant over white, and white is not completely dominant over red. And then now you produce what we call an intermediate color. Yes, or an intermediate characteristic. So this is called incomplete dominancy. Then you say that the type of barrier in speciation that split the single population into subpopulations. We call it the geographical barrier. Yes, we call it geographical barrier. Yes, remember we said that there was one population of, remember you have to apply when it comes to explanation of the speciation. There was one population of rats. You understand? The population is divided into two by a rock. So a rock becomes a geographical barrier into two subpopulations. So it divides this population of rats into two subpopulations. So how do you call structure which split this population into two subpopulations? We call it what? We call it geographical barrier. Yeah, correct. A reproductive isolation mechanism that occurs in flowers only. Flowers only. Obviously, flowers, they do carry out what we call pollination. So it's can call it, you are adapted to different pollinating agents. Yes, that is the word I wanted to modify. Pollinating, you are having a different pollinating agent. For example, the insects, you are specific to a specific insect with a specific shape of the beak. So we call it pollinating at different seasons of the year. So that is it. Then we go to the next question. They are saying that the genetic disorder resulting from a mutation caused by absence of blood clotting factor. A genetic disorder resulting from absence of a genetic disorder resulting from a mutation caused by absence of blood clotting factor. If you don't have blood clotting factor, it means that when you have a hole, maybe you have got something on your skin, the blood will ooze. So when the blood oozes, then it means that we call it hemophilia. Hemophilia. Yes. This is very common in ladies. Did I say in ladies? They are more affected because they don't have remember, hemophilia is caused by a recessive allele. So male are more affected by hemophilia, yes, than female. So we mostly find it in the males than in the females. Why? Because males they only have one X chromosome. So you can we will talk about it. If you find a question, they can ask you that why are males affected by this sex-linked disorder than females? You understand? As a result, you can say that males have two X chromosomes if a recessive allele appears on one X. Did I say males have two X chromosomes? Yeah. Females have two X chromosome. If a recessive allele appears on one X, it can be masked by a dominant allele on the other X. You understand? And then the person is still normal, but is a carrier. But if the males, once the recessive allele appear on one X, is carried on one X, automatically the person is what? Is the hemophilic. Then they're saying that the source of variation where individuals in the population has equal chance of mating with other. The source of variation where any individual in a population has equal chances of mating with other individuals. So how do you call that source of variation? So we say that we call it random mating. We call it random mating. Then evolutionary theory proposed by these gentlemen. Remember, we usually talk about three of them. We talk about Lamarck's theory, Darwinism, Darwinism basically is also being represented by the gradualism. And then we have punctuated equilibrium. So punctuated equilibrium, which occurs rapidly. It has periods of long periods of no change with rapid periods of change, meaning that they are alternating with one another. So it is the punctuated equilibrium. So you guys, yeah, we got the next question, which is 1.1.3. They are saying that a component of nucleotide found in DNA, component of nucleotide found in DNA and RNA. They're saying DNA and ribose sugar. No, this one is found. This one is found only in RNA, but they said DNA and RNA, so the answer becomes A only. A only. A only. Yes. They're saying that segment of DNA that code for a particular trait. This one is the segment of DNA that control a particular trait. So we call it a gene. It's A only. Advantage of bipedalism, wider view of the surrounding climbing trees. A only. Okay. Remember that you must always say A only, B only, or A and B, and then both A and B or none. So please make sure that you have this word only, 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 only. Yes. So we go to the second question, which is 1.4. The diagram below. So once they bring such diagram, they can ask you describe crossing over, draw the structure of a chromosome after and before. They can ask you a lot of questions here. They can talk about the karyotype. They can talk about Darwin's. They can talk about trisomy. Trisomes. They can talk about the trisomes. That is Down syndrome. Yeah. So what are they asking? So you guys, I always tell you that once you find this question, try to rotate it so that it looks like this, so that this and this, they are on the sides. Okay? So once in the side, you can see that this is the middle. So if you look at them, then it means that this is metaphase. And then once they are like this, because these are the spindle fibers, they are radiating like that. They are supposed to be in sides. So once they are like that, then it is easy for you to make it simple. So since it is one chromosome per, if there was another one here, then it would have been meiosis 1. But since it is only one chromosome, then it means that is meiosis 2. So now we know that that is yeah, so now we know that that is which diagram 1 or 2 represents meiosis. Diagram 1 2 represents meiosis and then represents mitosis. So this one looks meiosis, diagram 1. Diagram 1. And then diagram 2. Why did they bring this question? Anyway, I will say that it's diagram 2. Do you know why? Because here there are no variations. Here you see that the chromosomes, they are varying due to the crossing over. But here I don't see any variation. So it could be mitosis. So I'll conclude it like that. Name the process that occur in the cell division, diagram 1, that resulted in the you see, it is crossing over. So I will say that crossing over. Crossing over. In which phase does the process mentioned above occur? It occurs in the prophase 1. Occurs in prophase 1. Name the type of cell in which the cell division occurs. Name the type of cells in which the cell division, diagram 2. Oh, diagram 2. Remember I said this is mito. It looks to be like mitosis. And mitosis occurs, where does it occur? In the mitosis occurs in the somatic. Somatic. Use this color. Somatic. Somatic cells. It occurs in the somatic cells. Then they're saying that state the diploid number of chromosomes in the diagram. Diagram 1, diploid number of chromosomes. diploid number of chromosomes in diagram 1. Did I say it's a human cell? Didn't say anything about human cell. So it's going to be diploid number. It's going to be 4. And also this one is going to be 4. So, state the number for the cell. Now in diagram 2, remember, if this is mitosis, let's analyze it like this. If this is mitosis doesn't change the number of chromosomes. So if it is 2, they're gonna be 2 in meiosis in from the beginning until the end. So they're gonna be 2. So if they're saying diploid number, and here I'm seeing 2, so it means that this is not 4. This is gonna be 2. While this one, since this one it looks to be metaphase 2, then it means that in metaphase 1 there were 4. So there are 4 here. Like this. There are 4 here, while here there are 2. Why? Because in mitosis, the number of chromosomes don't change, while in meiosis, the number of chromosomes change. And if you look at it here, this is meiosis 2. That means that in meiosis 1, the number of chromosomes divided. So since these are 2, so in meiosis 1, then it means that there were 4. And give the number of chromatids in diagram 1. What I see. So it's 1, 2, 3, 4. So remember, each chromosome is made up of 2 chromatids. It's 1. Check. It's this. Then it's that. So it's this 1. So if I have 2 chromosomes, so it's going to be 1, 2, 3, 4. So the answer here becomes 4. In this bird species, dark feather color, capital D, is dominant over light feather color. Okay. And then long tail is masks the expression of the short tail. Okay. They are saying that the square below shows the possible results when the gametes of the parents were crossed. Okay. So if they are crossed, they are saying that name the type of cross shown in the diagram above. So this is a dihybrid because we are considering 2 characteristics. Yes. All right. Here you are seeing 4 of them, but those are not. If you are saying that size, you have big or small. So that is one character, which is size. Your body size. So that is one character. So you have to understand that concept so that you don't get it wrong. So they're saying that they name the type of cross represented in the Punnett square above. So we say that is dihybrid cross. They're saying that write the phenotype of the offspring. Right, the phenotype of the offspring. Just a moment. Write the phenotype of the. Just a moment. Okay, the phenotype of offspring X. It is easy. I go with this and this because that's where the cross is gonna be. Capital D, capital L, small l. Yes. Then they are saying that the genotype. Right. State the genotype of two parent 2. Oh. This is parent 2. So how do we get the genotype? So what I do, did I say genotype? They're saying genotype. So let me first identify the phenotype so that I'm able to identify the genotype. So the phenotype, I'm gonna say capital D, the first letter. Do I have any small d? Yes, I have a small d. You see? So small d. And then capital small l, and then even this is small l, so it means that it's capital D, small l, small l. Yes. So this is the genotype. Capital D means what? We come back and then find out. Capital D is dark feather. And then small l is small l, small l, small l, small l, small l. Mhm. Long tail masks the expression of the short tail. So this is small l. So it means that it's gonna be dark feather with short tail. Yes, so that is going to be the most correct answer. Mm, yes. Then they are saying that the dominant, the type of the type of dominancy, the type of dominance in tail length, with tail length. One is completely dominant over the other. So it means that is complete dominancy. So there I'll go with the complete dominance. Remember, if complete dominance, if one allele is completely dominant over the other, for example, light skinned crossed with the dark skinned, then you produce a kid of dark skinned, then it means that dark skinned is much more dominant over the light skinned. Tall crossed with a short and then it produces a short, then it means that tall is much more dominant over the short. Then you can look at any character and then you see what you get as a result could be that the other one which is not expressed was masked by the dominant allele. Yes. So which parent, one or two, has the heterozygous genotype for these characteristics? Which parent, one or two, has the heterozygous genotype for both characteristics? They're saying heterozygous. I see it is the parent one. You know why? Let's find out. Let's see here. Because here I have capital D, I write the capital D. Do I have capital D again? Yes. So I cancel it out. Do I have a small d? Yes. I have small d, so I write small d. So I don't write the D again. So I have capital L, capital L. Do you have another capital L? Yes. But do I have a small l? Yes. So I would go with a small l. So that is heterozygous for both characteristics. But if you look at parent one, you have capital D, capital small d, yes, small d. Small l, do I have a capital L? No. So I have to go with a second L. So you see that this is homozygous, recessive, or this is heterozygous. Well, this one is heterozygous, and also this one is heterozygous, because this is a capital L, and then this is a small l, due to that. Then they are saying that name two Mendelian principle that describe the separation of allele during the gametes. So here we can talk about Mendel's principle of segregation, and then you can also talk about Mendel's principle of independent assortment. We talk about the segregation, and then you also talk about the Mendel's principle of independent assortment. And then that makes us to get 50 out of 50. Who got 50 out of 50, or who is willing to get 50 out of 50? And if you got them, thank you so much. You are a star. So question number 2 is saying that, okay, question number 2 is saying that the diagram below shows different processes involving DNA molecule. So you have process A, you have process A. So it's question 2, you have process A and then process B. They're saying identify process A. If you look at here, we see that process A, you're forming DNA. So this is DNA replication. While DNA replication. While process B, we see this is the protein synthesis. Yeah, basically we see that this is transcription. So which part of protein synthesis is transcription? So process B is transcription, while process A is DNA replication. We can ask you a question: what's the difference between transcription and translation? What are the similarities between transcription and DNA replication? We can ask you the similarities between DNA replication and transcription. So you look at the steps that DNA replication occurs in the nucleus. Also transcription occurs in the nucleus. DNA replication, double helix DNA unwinds. It's the same story here. It also double helix DNA unwinds. If you look at the differences, you can talk about two strands they form the act as a template, while the other side you can talk about one strand act as a template in transcription. You can talk about two new DNA molecules are formed. This side you can talk about messenger RNA is being formed. So you look at the differences and the similarities. They can ask you those questions. They can talk about the differences between nucleotide of DNA and nucleotide of RNA. So they can ask you a lot of questions concerning about that. Name the organelle where process A and B occurs. Process A. So that is the nucleus. And then they're saying that basically we only have two organelles we usually talk about. We usually talk about the nucleus, then also talk about the ribosome. They state the phase in the cell cycle in which the event takes place. State the phase in the cell cycle in which event A takes place. We say that it occurs during interphase. So done with that, describe the event that occurred during protein synthesis after R molecule attaches to the ribosome. So if you are talking about R is a messenger RNA, so they're asking you to describe the process of translation. So you describe it. You must have the distinction material book. You'll be able to answer all these questions simply. Maybe you can say that according to the RNA anticodon, the transfer RNA with the complement. According to the RNA codon, the transfer RNA with the complementary anticodons will bring the required amino acid to the ribosome. The amino acids are joined together by the peptide bond to form a required protein. So basically that you can explain it, or specific amino acid is going to be brought to the ribosome when the anticodon of the transfer RNA matches with the codon of the messenger. You understand? The anticodon, the amino acids are joined together by the peptide bond. The bond is peptide. Don't make a mistake and say polypeptide bond. Zero will give you a zero, and that zero will have ears and eyes. Please don't do that. Yes. So the bond between the amino acid is called the peptide bond. Yes. Then they're saying that to form a required protein. So you explain as long as you talk about all the marking points which we are looking for. Describe the event that occurs during. Okay, we are done with that. Explain one functional relationship between DNA and molecule R. What is the functional relationship between DNA and the molecule R? Basically, molecule R contains the coded message from DNA so that the proteins can be manufactured. That's what they are looking for. It has coded information for DNA so that that thing can be processed, can be formed. Yes. The proteins. Then they are saying that explain the function. The table below shows the amino acid between DNA and the molecule R, which is a messenger RNA. They are saying that use the table below to determine the codons. When you talk about the codons, remember we have the codes which we find on DNA. We have the codons which we find on the messenger RNA. We have the anticodons which we find on the tRNA. Yes, so I've explained this before. If you check on our channel on Thunder Educ, then you'll be able to get all these questions and explanations about it. Yes, make sure that you get a distinction material book so that you're able to obtain these distinctions. Yeah, nobody, if you are using the distinction material book, you only don't get a distinction because you don't want. If you want it, is the distinction material book. The distinction is in your hands. All right. Then they're saying that write the triplet bases. Okay, triplet 1, 2, 3. So if you look at 1, 2, 3. So they are saying write the codons. So if this is DNA, then it means that I'm going to have to change it into a messenger RNA. So T goes as TGC. and then you have TAC, and then you have AGG. So it's gonna be T is gonna go with A, and then C, then G, and then it's gonna be A, it's gonna be U, it's gonna be G, then it's gonna be U, it's gonna be U, it's gonna be C, it's gonna be C. So basically this is what we are looking for, and that is what you are supposed to write. Yes. So you get your 2 marks there. How do they mark? I don't know, but they supposed to be like that. Determine the amino acid for DNA based triplet that is complementary to this. Guys, we always ask you these questions, and the mutation. Did they ask you? They didn't ask you about the mutation, but we always ask you about this. Determine the amino acids for DNA triplet that is complementary to TGC on strand 2. Determine the amino acid for DNA-based triplet that is coded to TGC. Strand 2 is DNA, and what is here is also DNA. So since they are giving you DNA and they are looking for DNA, then definitely we are not changing anything. So it's going to be TCG. Let me clear it up. TGC. So it's going to be cysteine. So that's what is going to be the answer. Let me check properly. Yes. Cysteine. Determine the amino acid for. Let me first clear it up so that I don't want to make a small mistake, and then yes, they're saying that determine the amino acid for DNA the DNA base, the amino acid for DNA base triplet. Okay, that code that is. Oh, you see, that is complementary. You see, because this thing was covered. So you see. Let me just do like this. Yeah, guys, make sure that you're always keen. They're saying complementary. So this is complementary. This is C. So what is the complement of this? T goes with what? Goes with A. G goes with what? G goes with C. C goes with what? With G. You understand? With the C, né? This is C, it goes with the G. So it means that this is still DNA. It's still DNA. It's not messenger, because what is given here is DNA. So it means that what I'm going to say, that this is just they are looking for the complementary. You understand? So since they are looking for this, so I'm going to look for this. So it's going to be ACG, which is threonine. So that is the correct answer. So you guys, you see these questions, né? You see, even me, I was gone because I couldn't see this because of the words which are there. So make sure that you take note of these questions. Né? Don't just give the answers and then you just go like that. Please make sure that you read these questions, because a small mistake like this makes you to get out of the what? Don't finish the paper and then you sleep. Doesn't make sense. It is like you are making the examiner to be a fool. Yeah, how can he set a paper for 2.5 hours when you finish it in 1 hour? You are making the examiner a fool. Yeah, when you don't know what you're setting. So guys, don't make us fools, né? Yeah, don't make examiners fools. Okay, we are saying that name the type of mutation that caused Jacobo's syndrome. What is Jacobo's syndrome? Karyotype below. Remember that karyotype is the type of the nucleus. Karyo means nucleus, and type means the type. Which type of nucleus do we have? Of the male suffering from Jacobo's syndrome. What's Jacobo's syndrome? We'll find out. Where is it? Name the mutation that occurred that caused Jacobo's syndrome in this image. I see here on chromosome number 23. Is this a male or a female? Is It's a male. Why? Because I have X and 2Y. But anyway, I'm not going to talk about that too much. I'm just answering a question which they said, and then yeah. So in most cases, we don't bring these questions which we bring the one which has the trisomy is on chromosome number 21. The one which were like this, they are being removed from the examination guidelines. So if they are brought them, let's answer them the way they are. Yes, but it's not supposed to be. Let me just say it like that. It's not supposed to be in this format. Anyway, yeah, following the examination guideline, that is my opinion. Yes. However, they also had their opinions. Why did they say it? Maybe they want to say it about something. All right, we continue. However, you can use the same principles used here to answer these questions. The good thing, they're not asking you what is it. They're just application question which you can apply and then explain. So use the concept of Down syndrome, and then you are able to obtain or transfer that knowledge to this question. Maybe that's the instance for them setting such question. They're saying that for this individual, determine the number of autosomes. Remember, autosomes are the chromosomes which are not they are not sex. So I'm going to have to count out is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. Guys, always count them, because sometimes there is one where there is 3 of them. So autosomes, there are 22 pairs. Or I can say they are 44. Yes. Then gonosomes, we see them, there are three chromosomes. Yes, I can't say 3 pairs because they are just 3. 1, 2, 3. So these are 44 chromosomes, not pairs. Yeah, 22 pairs of chromosomes or 44 chromosomes. Use letter X and Y. Write the gonosome of this individual with Jacob's syndrome. So we see that this is X, the one which is long is X. Let me clear it up so that you're able to view. This is X, this is X, this is X, and then this is Y. So basically that is it. Yes, they're saying describe the way in which the karyotype of the person with the Down syndrome will differ from the one mentioned above. Remember, Down syndrome, the trisomy is basically happening on chromosome number 21. Like the 3 chromosomes will appear on chromosome number 21, while this one it appeared on chromosome number 23. You understand? Yes. So basically, the trisomy 20 Down syndrome, it occurs in the autosomes, the autosomes which are non-sex chromosomes, while this one occurs in the sexy chromosomes, which we call the gonosomes. So I think that's it. Explain how the error during the gamete formation in meiosis 2 could result in the Jacob syndrome. Then it means that if you look at this, what happened is this could be for a female or it could be for a male. But what happens? It means that during anaphase, yes, the chromosomes for the chromosome pair, like 23, want pair. The 23rd chromosome is supposed to, for example, this one is supposed to split like this. But now, it is in metaphase. So during anaphase, they are supposed to split so that they look to be like this. So they are moving in opposite poles. Yeah. But now what happens on this gonosome? What will happen? This chromosome, which is the gonosome, for example, instead of this one, instead of them, instead of splitting like this, both of them are going to go to one side, meaning that leading to this other side having nothing. So we're going to result in this cell having two of the gonozome. You understand? And then this side, there's nothing because nothing went there. It's only have these orosomes which are there. So it means that if this was a male, no, it was a male, yes, is going to have X. You understand? Two of them, X. So if it was a male, it's going to have Y, Y. And then this one is going to have an X. Zero. Nothing is there. So there is no gender there. So if this cell fertilizes a female ovum, the ovum, the ovum, yes. So what is going to happen? It means that the ovum is going to bring X, so that now this individual is going to have one X and two Y chromosome. So that is what is supposed to be. So basically here, non-disjunction occurs as the failure of the chromatids fails to separate at position 23. So that's what we talked about. And then he said that during in anaphase 2, because this is, they are saying in meiosis 2, a non-disjunction occurs in anaphase 2. During anaphase 2, the sperm cell will have an extra chromosome, you see, due to the failure of them to separate. When it fertilizes a normal ovum, yes, the individual is going to have one X and two Y chromosome. So, which is what we call the Jacobo's syndrome. But I talked about it that in most cases, we don't usually set this. I think they were just setting it to link it to Down syndrome. We like it to bring it to Down syndrome. And then if you see how I was explaining it related to Down syndrome. But instead of writing 21, chromosome number 21, I used the chromosome number 23. Yes, so that is it. And then we are saying that 2.3, they're saying that state the phenotype of anger. Okay, here we are having this and this. So to know the phenotype, you need to know what is the kids. How many generations? There are two. 1, 2, 3. So there are 3 generations. How many kids are for James and Anga? Is 1, is 2, is 3. Okay. Someone is going to ask, how is it 3? Because this shows that it gave rise to that and that and also that. So this they just got married to those kids. So they're saying that the genotype of this, so if this is blood group A, they produce blood group AB. Definitely this is blood group B. You understand? So that this person, since this blood group B, blood group, yes, they produce this. They produce this. Why not blood group AB? AB. Because if there was blood group AB, then it means that the genotype is going to be like this. There is no way how they're going to produce handle. So it means that this one is blood group A, heterozygous, and this is blood group B, heterozygous. So that this and this, they combine and they produce tandem. Yes, so it means that Anga was blood group B. Then the genotype of individual that plays codominance, which is codominance, that is blood group this AB. So the genotype, so definitely it's going to be A. B, because they're looking for the genotype. They're saying that give the number of individuals with homozygous recessive allele, that is blood group O. That is blood, but they're saying give the number. How many are they? So it's 1, it's 2. So it's going to be, in this answer, this question is going to be 2 of them. Explain why the future children of JD and Lisa could display different blood groups compared to their current children. Lisa, JD and Lisa. I have to look for JD and Lisa. So since JD and Lisa, this is A, this is B, this is A. And then since these ones were like this, then automatically JD was a small letter. And then to produce a different blood group, yes, to produce a different blood group, then automatically even A must be heterozygous. Oh, this is B, ne? JD is B. And then so that their children could be, when this small letter and small r, when they combine, they're able to produce blood group O, meaning that the JD will produce will pass a recessive allele to the offspring, the same way Lisa is going to pass the recessive allele to offspring to produce blood group O. But I need to find out what is the genotype of Lisa, which is blood group B heterozygous, and also JD is blood group B heterozygous, and also Lisa is blood group A heterozygous. So you see that to obtain any answer related to this, make sure that you look at the babies they have done what they have produced. Then you are able to obtain those blood groups. Yes. So we go to the next question. Use the genetic cross to show the percentage possibility of beta and the Oneka having children with blood group O. So I have to go back and look for beta. Where is beta? And then Onek, what? Bela, and Onek, the same genetic cross. So you have, since they are on the second generation, I will say that P2, F2. And then they are saying that the phenotype, phenotype. I told you that this 1 mark is obtained free, mahala. So genotype, I come the question. They're saying that Baila, blood group what? Blood group of blood group what? Baila is blood group AB of blood group AB crossed with Onita of blood group what? Of blood group of blood group O. Blood group O. So when you look at the genotype, blood group AB looks like this, while crossed with Onika, which is blood group O, small I. Then they form meiosis. You have to write all these points, and then you say that A is gonna be B, is gonna be small I. So these are gametes. So we call them gametes. I say that don't leave them naked because they're not at the beach. So they are supposed to be dressed up. So after that, you can use the Punnett square, or you can just do the crosses. So let me just use the crosses so that I'm able to be a little bit faster. That with that is going to be A, small I, and then now this with that is going to be A, small I, then this B, this with I, that's going to be B, I, then that with that is going to be B, I, small I. So these are offsprings. So we see that these two blood group A, and then two blood group B. So we don't have any chance for someone to produce blood group which is O. So it means that 0 out of 4 times 100, so it gives me 0%. So the probability to produce this kid who is like this is going to be 0%. Then we go to the next question. This is genetic engineering. We can ask you genetic engineering questions. We can also ask you about stem cells. Yes. So the type of cell produced by meiosis that is extracted from the donor kernel. So we look at this. This is a somatic cell. This is a somatic cell, and this is the ovum. That's a gamete. That's the gamete. So somatic cells are always diploid, while this one are always haploid. So that now, because we're looking for the animal which looks like this, so I'll keep the nucleus here. Check, this looks like this. So it means that we use the genetics of this, which is the somatic cell. So it means that I'll keep the nucleus here. I throw away the shell. Here, I throw away the shell. The nucleus, I keep the shell. That's why you see the nucleus here looks like the nucleus which came from here. You see? And then if you look at the shell, looks like that of thick and thick. So now when you have this, we use what called a mild electric shock to combine the nucleus from here and then the shell from here. So we combine them together using the mild electric shock. Then now these embryos are being developed, meaning that they have the genetics of this. But because this embryo, this cell looks like a zygote, because remember zygote, it has a sperm, and the sperm only the nucleus fuses, and then the ovum. So this nucleus of the sperm goes into the nucleus of the ovum. So outside is the ovum and the inside is diploid. So since this is a diploid, yes, and the nucleus outside here, this is the membrane we used, looks like this. So this cell looks like a zygote. So it's not a gamete. Why? Because this is 2N, it's a diploid. So now it undergoes mitosis to form a ball of cells which develops into embryos. So these are being implanted into surrogate mothers, and then each surrogate mother develop, remember these are just stem cells, the function of stem cells, then they develop to form mature babies, and these babies are being released. So that is it. So name the type of cell produced by meiosis that is extracted from the donor cell. The type of cell produced by meiosis that is extracted from the donor cell. So we're gonna call it a gamete. Yes, basically it is ovum because it is a female. They're saying the organ where implantation takes place, it takes place in the uterus. Uterus. They're saying the cattle counted to be 63% of 395 different cloned animals in the particular country. Calculate the total number of cattle that were cloned this country. So this one is simple, 63,3 divided by 100 times 395. So the answer you get, that is the answer. I don't have a calculator here, so you can calculate and then you find what is the answer there. Then they are saying that explain how the process of cloning will affect the variation of the cattle. Obviously, cloning result in the identical organism. So it means that the variation is going to be reduced. So it's going to be reduced variation. Suggest the different characteristics desired by human that influences the cloning of the 3 characteristics desired, like meat. You can talk about meat. You can talk about resistance of diseases. You can talk about meat, resistance of diseases. You can talk about milk. So all those can be used to clone, be reasons to clone these organisms. So this question is also 50. So if you got 50 out of 50, congratulations, you are a star. Let's go to question number 3. Go to question number 3. They are saying color blindness is a sex-linked genetic disorder that is characterized by the genetic defect of on X chromosome. The moment you say X chromosome, then it means that you want to use X and Y. They're saying that the people with color blindness cannot differentiate between certain colors. The table shows, okay, according to the table, which region has the highest or has the lowest percentage of male with the color blindness. So if you look at it here, we shall see that this is the answer. So it becomes Sub-Sahara Africa. This is just a question which is an application. It can be changed. It's easy to answer. Yes, suggest one reason why the results in the region identified in the question might not be correct. Why should we? Why should do you think? Maybe there are no proper facilities which are used to test. Yes, maybe the facilities are not there. Maybe there is lack of reporting of some cases might not be reported, and then so the information could be wrong. So things which can make the results not to be right, or not everything which has been collected could not be right, or some information might be missing. Yes. So the prevalence color of color blindness in the female is 0.5 globally. Explain why female have a lower chance of being colorblind. So this one is talking about, this is what I explained, that females they have less to be affected by hemophilia. This is a sex-linked character. You understand? Yes. Why? Basically, females have two X chromosomes, so they will require 2 recessive allele for them to be color blind. So if one recessive allele is appear on one X, it can be masked by a dominant allele on the other X, and the female is still normal. However much is the carrier, but it's still what? Normal. So females will require 2 recessive allele on the X chromosome for them to be color blind. You understand? Therefore, it means that even if one recessive allele appear on X, then it can be masked by a dominant allele on the other X, and still the individual is still normal. Then you draw a bar graph. You guys, this is question you must be knowing. Make sure that the bars are the same size. Make sure what you have here This is X, this is Y, so this one goes to X, so it comes here, and then these numbers, they go here on Y. You can't have words here, never on Y axis, no, they must always be on the X axis. Then you put the caption, make sure that the caption, you put them properly. Make sure that what you put here, you put units if the units are there. In this case, the unit is percentage, so the percentage must be there. Countries, region, is basically, these are countries, so you can put in brackets a region. So it can be something like a unit, and then it must be indicated there. And then make sure that these bars are the same, the spaces between the bars are the same. Then you'll be able to obtain all the marks, the 6 marks they have given you. Then we go to question 3.2. 3.2 is a little bit easier. They're saying the diagram below shows the scar of hominid ancestor. Yes, the hominid ancestor, and they used tools as their evolution. So they are looking at the evolution, the tools they used as their evolution. Okay, they're saying state the line of evidence for human in evolution. In the diagram, state 2 lines of evidence for human evolution in the diagram. So in this case, we see that we have the tools, and then we have the fossils. So remember, when you're explaining, you don't say tools. Remember, the evidence is cultural evidence. Example of cultural evidence is use of tools. So the answer here is cultural evidence, and then here number 2 is fossil evidence. Then they're saying that name 2 Australopithecus africanus species found in South Africa. We talked about them: Taumuchad, Littlefoot, and Mrs. Place. So this one are easy to identify. Identify the type of tool used by species with the largest cranial capacity. So if you look at the tools they use with the largest cranial capacity, so we see these are the tools they used. Then they're saying that how many genera are shown in the diagram. So if you look at the genera, we look at the first, like the surname. So we see the Homo is 1, is still Homo, is still Homo 2, and then this is 3. So there are 3 genera which they shown in that diagram. They're saying that based on this diagram, the Handyman species was in existence 1.8 million years ago. Write the scientific name for handyman. That is Homo habilis. So handyman, remember, is the first man to use tools, so we call them Homo habilis. Provide a reason why species named above is regarded as handyman. It means that it's the one which first used the tools. So I've given the answer. Explain why the evolution of large cranium in human is associated with the development of tools. Why is that the more the brain is developing is linked to the advancement of tools. Advancement of tools, these tools, they require thought. So it requires the brain which is thinking, the brain which is much more, how do I call it? Much more developed to think and develop these tools. So we can say the increase in the brain will lead to the increased intelligence. Actually, when you have a large brain, you're going to have more brain cells, which leads to the increased intelligence and then leading to the development of the complex tools. So increased brain means that you have much more brain, yes, which increases the intelligence, leading to the increased intelligence, and then this means that you're going to have to develop more complex tools. 3.3, what are they asking about? 3.3. Here is an extract you're supposed to read. And then we are saying that lactose intolerance refers to the ability to digest glucose, lactose sugar into glucose, and lactose tolerance individuals do not experience comfort after drinking milk, eating dairy products. Lactose. However, some individuals struggle to digest lactose due to the production of lactase enzymes, and then classified lactose intolerance. Such cannot be. So scientists investigated the influence of lactose intolerance gene mutation on the lactose tolerance in African population. Okay, this is very important. They're saying that they collected DNA, okay, of this. They're saying they were found, okay, in Tanzania, Kenya, and Sudan. And then they're saying that divided the participant into two groups, okay. They're saying that, okay, I'm seeing the groups gave the participant lactose sugar solution to consume daily. Okay, then ensure that participants ate the same diary during the investigation, measure the blood glucose before and after. Okay. What is the independent variable? So what determines this concept? So basically, the independent variable is the lactose intolerance gene. This, because I say investigate the influence of this on this. So it means that this is the independent while this is the dependent. So lactose tolerance is the dependent. So this is done. Name the type of variation existed in the ability to digest lactose. So it is discontinuous. Either you digest or you can't digest. You can't try. No. You either do it or not. So it is two, you get your mark there. It is discontinuous variation. Then they're saying that state way in which the scientists ensure the reliability of the investigation. Remember reliability by increasing the sample size. So they said they ensured, meaning that they used 470 people. Yes. The individuals were from different ethnic groups. You understand? We see them there. We see that for different area of the what? Sorry, guys. Alhamdulillah. So we can talk about all of that. State the factor that should be kept constant when the lactose solution was consumed. Which factors? The amount of lactose which you consume must be kept constant. The concentration of lactose must be also kept constant. Why are scientists measure the blood glucose level before and after? Basically, they want to compare these results by developing what called a baseline. Yes. Okay, before it was like this, after it was like this, what caused what? That change is caused by what? So they want to find out the effect. So by developing what called a baseline. Then they're saying that describe how scientists could establish whether the gene mutation has occurred on this, whether a genetic mutation had occurred on lactose this gene. So basically the sequence of gene of DNA or of the participant on the chromosome, yes, should be determined. And then they have to determine it using the DNA sequence, you understand? And then after that, they have to identify the mutation of this gene, and then they compare it to the one which is lactose intolerance. If they see that there is a difference, then automatically it means that there was a mutation. So they have to first identify the one which is tolerant and the one which is intolerant. And then after that, they must compare. And then they see that, oh, the one which is intolerant, maybe the mutation occurred. If there is a difference, then automatically it shows that there was a change in the sequence of the gene. So which means that it was a mutation. Then we go to the next question. Large scale overfishing. Okay, so remember much of the rate of discipline and humans in target fishing and port nets. So this 30% reduction of the size of faster maturation. Okay. According to the paragraph, which human activity has resulted in the... So when we bring such question, guys, make sure that you read the passage and then you read it at least 3 times so that you're able to obtain the information which is there. According to the passage, which human activity has resulted in the population of Atlantic codfish? Basically, if we see that there is large scale overfishing, then they are saying that explain the effect of GHR gene mutation on the survival of the chance of Atlantic cod fish. So basically, what is explain the effect? What is the effect of this? What is the effect of this? Yes, mutation on the survival. So we see that this mutation was useful. How was it useful? Yes, because it's basically reduced the size. Ne? So if the size is reduced, it means that the fish could be a little bit faster and then increase the chances of their survival, meaning that they are able to move very fast. They are swiftly moving, and then they are hiding. They're able to hide from the predators or from the hunters so that their population can increase. Use down theory of natural selection explain the evolution of small body fish, small body size, and the faster maturation rate in the Atlantic codfish. So what do we explain here? Remember, this is natural selection, so you can have to tell us that there is variation among these codfish. You have to tell us that. Then you tell us the variation. Which variation is that? Some had experienced mutation of on GHR genes that resulted in the small body and then making them to a little bit faster and then than others, and the others did not have that, meaning that the one which had the mutation, yes, they were able to be faster, you understand, and then others they didn't have that ability. So that is the variation which we are talking about. So basically when overfishing occurred, yes, so that is now the pressure which is being exerted to these fish. The codfish without the mutation, what happened to them, meaning that they could move less, so they were being hunted, they died in large numbers. All those ones with the mutation, they could move faster, so they survived in large numbers. You understand? So they reproduced also in large numbers. They passed the allele for this mutation to the next generation, and then after many generations, it means that the next generation will have a higher proportion of this codfish with this mutation, and then with a less proportion of those ones without that mutation. So basically that's what you're supposed to explain. Please don't confuse this natural selection with speciation, because in most cases students confuse this and then they end up losing the mix. They are saying discovery, there's so many passages. Discovery of fossil in remains of 87 million years of okay, scientists Europe understand the okay this call found in this transition has a transitional future similar to that of Africa. Okay, name the hypothesis of human origin that is being challenged in this passage. So we call it the out of Africa hypothesis. We don't have any other hypothesis. State the hypothesis mentioned above. Modern humans, the word modern must be there. If you don't put it, remember we have different humans, homos. All homos are humans, but the modern is the Homo sapiens. So you have to be specific and say that modern humans originated from Africa and migrated the rest of the world. Yes. Then they're saying that describe 3 characteristics of the skull that's where similar to that of these African apes. So foramen magnum at a more forward at a more backward position. Yes, it is in a more backward position. And then you can talk about large brow ridges. You can talk about they were prognanthus, meaning that they were prognanthus, meaning that more protruding jaws. Yes, and then you can talk about the rectangular palates. So the way how these apes were. Yes, they're saying that what is the name of the birthplace where most fossils of mankind were found? So this is cradle of humanity. So basically that is the paper, and since the paper was not that difficult, I see that you guys you will make it, and then we see that so guys, yes, we wish you the best in your exams, and we pray that you guys you pass you very well. I've not talked about the phylogeny. The phylogeny was there. Make sure that you do all those questions which are found in the phylogenetic tree at the back of the distinction material book, so that you'll manage the paper. Don't stress about it. You'll manage it. You'll pass it. It has been M.Saidi. I love you so much, guys. Thank you for loving Thunder Educ. And just know that we are here to see that you guys, you become responsible citizens who are not just citizens, but those citizens who are rich.